In this video, we will talk about Huntington's disease, as we have discussed that best examples of trinucleotide expansion are Huntington's disease, Fragile X syndrome, and myotonic dystrophy type 1. Let's move into details of Huntington's disease. 4p16.3 refers to band 16.3. On the short arm of chromosome 4, the numbered bands are used to specify the location of the genes found on each chromosome. Expansion of part of the short arm of chromosome 4 causes this disorder. Huntington's chorea shows anticipation. In these conditions, symptoms often become more severe generation by generation, or appear at progressively younger ages, called anticipation. Huntington's chorea shows anticipation when transmitted paternally. Gene product of Huntington's chorea is Huntington. Frequency of Huntington's chorea is 1 in 10,000 to 20,000 Caucasians. Now let's compare normal brain and brain of patient suffering from Huntington's chorea. Normal brain has normal-sized ventricles, but Huntington's chorea's brain has enlarged ventricles. Now let's discuss features of Huntington's chorea. These are slowly progressive neuronal death, characteristic Choreic movements, chorea or chorea occasionally is an abnormal involuntary movement disorder. The term chorea is derived from the ancient Greek word chorea, which means dance. So, involuntary jerky movements of arms, legs, face. Gait becomes unsteady, and speech unclear. There is insidious impairment of intellectual function and psychiatric disturbance, culminating in dementia. Dementia is not a specific disease, but is rather a general term for the impaired ability to remember, think, or make decisions that interferes with doing everyday activities. Advanced patients have difficulty swallowing and generally die of pneumonia, cardiorespiratory failure, subdural hematoma after head trauma or suicide, a pool of mostly clotted blood that forms in an organ, tissue, or body space. A hematoma is usually caused by a broken blood vessel that was damaged by surgery or an injury. It can occur anywhere in the body, including the brain. The average age of onset is 41 years, and the mean duration of illness 15 years. 5 to 10% of cases present before 20 years, with rigidity, slow clumsiness, and early progressive dementia, often with epileptic seizures. 80% of juvenile onset Huntington's disease cases show paternal transmission, and these have especially long repeat series. Now let's discuss etiology of Huntington's chorea. Normally, in the messenger RNA, repeat unit is CAG. Normal range is 6 to 35. This CAG codes for glutamine, but in deceased condition, the Huntington gene contains a highly polymorphic CAG Trinucleotide repeat series in an exon near the 5' end. In most patients, this is abnormally expanded. possibly due to slippage of DNA polymerase during spermatogenesis. Let's understand it. In the messenger RNA, repeat unit is CAG. Its diseased range is 21 to 220. Normally, CAG codes for glutamine, and the expansion is expressed in the protein as a polyglutamine repeat. This affects the solubility of the Huntington protein, causing it to precipitate out within and near neuronal nuclei, especially in the corpus striatum. Cleavage of the protein aggregate by the apoptosis enzyme, caspase, creates a toxic product that kills the cells, causing late-onset neurodegenerative disease. Huntington's disease is the prototype of a well-defined group of polyglutamine tract diseases, which includes several of the spinocerebellar ataxias and Kennedy disease. The spinocerebellar ataxias are genetically and clinically heterogeneous group of neurodegenerative disorders, characterized by a slowly progressive cerebellar syndrome in association with oculomotor, retinal, pyramidal, extrapyramidal, sensory and cognitive, or behavioral symptoms that vary with the affected gene. Kennedy's disease is an inherited motor neuron disease that affects males. It is one of a group of disorders called lower motor neuron disorders, which involve disruptions in the transmission of nerve cell signals in the brain to nerve cells in the brainstem and spinal cord. Now let's discuss the management of Huntington's disease. Development of Huntington's disease can be monitored by magnetic resonance imaging. Is a medical imaging technique that uses a magnetic field and computer-generated radio waves to create detailed images of the organs and tissues in your body. Most magnetic resonance imaging machines are large tube-shaped magnets moving towards treatment of Huntington's disease. Treatment is with medications to control choreic movements, antipsychotic drugs, and antidepressants. Caspase-specific inhibitors may be beneficial. Huntington's disease is an autosomal dominant disorder. In an autosomal dominant disorder, the altered gene is a dominant gene located on one of the non-sex chromosomes called autosomes. It means person needs only one copy of the non-typical gene to develop the disorder. Let's understand its pattern. If a diseased person having one normal and one diseased chromosome marries to the normal woman having both the chromosomes normal, this side is for father and this side is for mother. Normal chromosome from father and normal chromosome from mother in second child. Normal chromosome from father and normal chromosome from mother in third child. Deceased chromosome from father and normal chromosome from mother in fourth child. Deceased chromosome from father and normal chromosome from mother. So 2 out of 4 children are diseased. We can say chance is 50% that the progeny would be affected. So Huntington's disease is an autosomal dominant disorder. Let's discuss. Huntington's disease phenotypes. Phenotype is normal. If number of CAG repeats are less than 26, its features include stable throughout meiosis. Phenotype is normal. If number of CAG repeats are between 27 to 35, its features include permutation, unstable in paternal meiosis, reduced penetrance, Huntington disease. If number of CAG repeats are 36 to 39, its features include late onset or non-penetrance, full penetrance, Huntington disease. If number of CAG repeats are greater than 40, symptom starts from average age 41 years. Juvenile onset, Huntington disease. If number of CAG repeats are greater than 55, symptom starts before age 20 years. So, this was all about Huntington's disease. In our next video, we will talk about the Fragile X syndrome.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.